We present BioCicle, a tool for visualized taxonomic reports out of biological sequence comparisons. BioCicle aims to help bioinformaticians understanding taxonomic relationships between organisms by tackling two different analysis tasks. The first one summarizes taxonomic reports out of single query alignments, and the second one compares taxonomic reports out of multiple query alignments. I am first presenting you a single query taxonomic report for a protein sequence comparison. In this case, we are using the protein accession ID to generate the sequence alignments. However, it is also possible to upload a pre-generated comparison result in their usual format. The sequence we are going to compare is taken from a rat. The results are shown in a tree visualization, where each leaf represents an organism with which the sequence of the rat was compared. In the right side, you can see an icicle. Each leaf height is calculated considering the maximum score value for the sequences belonging to such organism, and their parent's height is calculated as the sum of the children. It is possible to filter the icicle leaves by using a score threshold. This threshold depends on the result's highest score. For example, if the highest score is 300 and the score threshold is set to 50%, only the leaves with a score greater or equal than 150 will remain. In this case, the highest score is set by the organism Rattus norvegicus. The second greatest score is set by the organism Mus musculus, which obtained only 42% of the rat's score. The second analysis task aims to support similarity detection for multiple comparison results. In this case, we are going to present a case study consisting of a set of 179 sequences. These sequences were obtained by sequencing a genetic marker, the 16S, over a single sample. The visualization consists in three main views: an icicle iteration to the right, a dendrogram to the left, and a set of small multiples on top. The iteration goes through the entire set of results. The small multiples present the taxonomic profiles for each of the results, and the dendrogram shows the taxonomic grouping of the sequences with which the dataset was compared. Considering the amount of nodes it can have, it is presented as a collapsible tree that dynamically expands its nodes according to the number of visible children. The nodes in the dendrogram are sorted following the number of results containing sequences with such node in their taxonomy. It is possible to see that there is a diverse distribution of taxonomies over the sample. However, yellow, purple, and orange tones prevail in the general side. If we filter by the most common order, gamma proteobacteria in this case, we can see that 88 out of the 179 results are still remaining. Also, most of the results present purple tones over the entire tree, which leads to conclude that a great amount of sequences with high scores in the remaining 88 results belong to such order. After repeating the filtering process with the next two most common orders, alpha proteobacteria and delta proteobacteria, it is possible to see a similar behavior as with the gamma proteobacteria. The remaining results for both filters have common tones between them: yellow for alpha proteobacteria and green for delta proteobacteria. As a result, with this simple process, it is possible to conclude that out of this dataset of 179 sequences, a considerable amount of results have significant scores with organisms belonging to the order Gamma proteobacteria. Also, some of them have similarities with organisms belonging to Alpha proteobacteria and Delta proteobacteria. BioCicle is a tool that aims to support bioinformaticians in taxonomic report analysis using straightforward visualizations.